My name is Zhe Han, and I am associate professor at Children's National Health System. I'm a Drosophila geneticist. We use Drosophila, the fruit fly, as a model to study human disease. I've been doing this fly modeling of human disease for almost 20 years since I was a graduate student. We just initiated a new study to make Drosophila model for APOL1 nephropathy, which is related to African American specific kidney disease. People noticed about 10 years ago that African American has higher incidence of developing kidney disease. 6 years ago, a group led by Martin Pollock from Harvard identified the gene APOL1 to be the reason of this higher risk of kidney disease. And they identified two risk alleles called APOL1 G1 and G2 to be responsible for this higher risk of kidney disease. The reason that so many African American carries this particular risk allele is because the risk allele, we call it risk allele for kidney disease, but those variations actually are beneficial for the African people because it offered a protection for African sleep sickness. The number of people who carries this risk allele is very high. Among African Americans, almost 20% of them carry two risk alleles of APOL1. 42% of African Americans carry at least one allele of this gene. So almost half of African American has one allele. One allele does not lead to higher risk of kidney disease. However, two allele will lead to up to 30-fold of more risk of kidney disease. So this is what makes this APOL1 nephropathy such a significant health issue. The risk allele of APOL1 will also affect the kidney transplant, because we don't really know the mechanism of APOL1 nephropathy. So we do not have any targeted drug to treat this disease. So right now, kidney transplant is the only option. However, when people receive transplanted kidney, many of this kidney, if it's coming from African American, it also carries this risk allele. There already been a recent study showing that transplanted kidney that carrying this risk allele will last much shorter than a kidney that do not have this risk allele. So this is make it even a bigger health issue. One very interesting thing about APOL1 is this gene is highly human specific. So only human and certain chimpanzees have the APOL1 gene. So people thought this gene was very exclusive to human, and so it's very hard to study. However, this is why we developed the transgenic fly model for APOL1. Although Drosophila, the fruit fly, doesn't have this gene, we can put this human disease gene in the fly, particularly in the fly kidney cells, and see what happens. We're the first team to do the fly model for APOL1 nephropathy. Recently, we presented this study at the ASN annual meeting in Chicago. This meeting attracted more than 16,000 attendants from over 100 countries, and our APOL1 study attracted a lot of attention because the APOL1 nephropathy is becoming a very hot topic in the kidney field, and people are realizing how important it is. In fact, my colleagues has been generating the APOL1 nephropathy model in other model systems, and we exchange some idea, and we want to work together to identify the possible cures as soon as possible. Drosophila cardiac nephrocyte, previously called pericardial cell, function as a filtration kidney in fly. It's a combination of mammalian podocyte and renal proximal tubule cells. We have two JSON papers in 2013 to demonstrate that. We also developed a genetic screen system to looking for genes that are essential for renal functions in fly. To really understand why APOL1 risk allele causes disease is really important not only for those who carry two risk alleles and develop kidney disease. It is also important for those just carrying this risk allele because they will pass this risk allele to their children. The next logical approach is to use a preclinical model to figure out why APOL1 risk allele cause kidney disease, and to use the model to identify a positive drug, maybe through a drug screen approach, and identify the targeted drug for APOL1 risk allele. One additional important thing is APOL1 nephropathy has been closely linked to HIV infection. So people who have 2 copies of APOL1 risk allele may not necessarily develop kidney disease. And again, people with HIV infection may not necessarily develop kidney disease. But if a patient has this risk allele and a positive for HIV infection, they're almost certain to develop kidney disease. And the reason is also totally unknown. I'm collaborating with Dr. Patricia Ray on Children's National Health System to identify the specific interaction between HIV risk allele and HIV proteins. So we're actually looking at every single HIV protein, and we believe one of the HIV protein may physically interact with APOL1 protein to produce pathological effects. Another follow-up approach to address this question is to identify the so-called second hit of APOL1 nephropathy. Because we know people who have 2 copies of APOL1 risk allele may not always develop kidney disease. So people believe that there is a second hit. When that second hit is also mutated, then the patient will develop kidney disease. By doing a Drosophila genetic screen, which we are currently doing right now in my lab, we aim to find the second hit, which when we silence another gene at the presence of APOL1 risk allele will give us a more prominent kidney defect. The APOL1 risk allele itself is a driver for kidney disease, but it may need another driver which either makes the APOL1 risk allele more potent or create another deficiency which synergizes with the APOL1 risk allele and eventually break the threshold of developing a kidney disease.